All right, now let's talk about section 3 of chapter 9, fermentation. So, fermentation takes advantage of the first step of cellular respiration, glycolysis. And as is pointed out in the book, the glycolysis is a relatively simple process and has some benefits in that it can quickly make ATP, okay, and make it available to do work in the cells, and it does not require what? Does not require oxygen. Okay. Um, now, um, without oxygen, you can't run the electron transport chain because, as we saw, oxygen is the final electron acceptor in the process forming water, and so that can't happen without oxygen. So electron transport chain can't happen, and the Krebs cycle doesn't happen. So essentially, cellular respiration doesn't happen. So you're obliged to do uh, fermentation if you can to generate some ATP. All right, so let's talk about the different ways that organisms can operate in regards to oxygen. So, of course, you can be anaerobic and not use oxygen. Some organisms are what we describe as obligate anaerobes. That is, they do not like having oxygen around at all. In fact, to them, oxygen is basically toxic. Thus, they are obliged to be anaerobes. A good example is Clostridium botulinum. This is a microbe that causes a type of food poisoning known as botulism. The microbe thrives in when people do home canning. If they're not sterilized properly and Clostridium botulinum is present, they can thrive in the low oxygen or no oxygen levels in those cans, and they produce what's called botulinum toxin, which if you ingest enough, that it can kill you. It's also the toxin that's used for Botox at low doses in your skin. It can basically sort of temporarily paralyze the muscles of those cells under the skin. Um, you can be a facultative anaerobe, that is, you don't use oxygen, but oxygen is not toxic to you. So if it's around, it's not a problem. It's just that you don't use it. E. coli, Escherichia coli, I'm sure you've heard of E. coli, is an organism that is a facultative anaerobe. It doesn't use oxygen, but it's not toxic. E. coli, as maybe you know, live in the gut of animals, in particular mammals, and we have E. coli in us. So you can also be an obligate aerobe, which we are basically. That is, you have to have oxygen in order to survive. In the microbial world, a good example is Mycobacterium tuberculosis, a bacterium that causes the disease tuberculosis, which is an infection of the lungs, and obviously there's lots of oxygen in the lungs, and these guys make use of it and can thrive quite nicely under those conditions. So you're obliged to have oxygen. They have to have it. All right, but let's look at fermentation, this condition that occurs in the absence of oxygen. So essentially, you notice we have glycolysis here, the first step of cellular respiration. We take glucose, break it down into pyruvic acid or pyruvate, generate some ATP and these electron carriers, NADH. But now, without oxygen present, again, you can't do the Krebs cycle electron transport chain, so what does the cell do with that pyruvic acid? Well, there's two types of fermentation. You can do what's called alcohol fermentation, in which the pyruvate is converted into ethyl alcohol, or you can do lactic acid fermentation, in which the pyruvate is converted into lactic acid. And those are two separate strategies that organisms do. There aren't organisms that do both. They'll either do one or the other. All right, so let's look more closely at alcohol fermentation. So in alcohol fermentation, notice we're converting the pyruvate again into ethyl alcohol we're also producing some CO2, and notice that we have to make use of those electron carriers that we just made. Okay. This is a process that's used. There should actually be a arrow right here. I don't know why there isn't one. Pyruvic acid plus NADH makes alcohol, CO2, and NAD plus. So as you're probably aware, this is a process that's used widely in the production of alcoholic beverages. Um, and of course, the bubbles, the foam on the top of this beer that's fermenting is coming from the CO2 that's being produced. We also make use of alcohol fermentation in bread, the production of bread and baking of bread. And what's essentially happening when the bread is rising is fermentation is occurring because you're using, in that case, well, and in brewing as well, yeast is the organism used to do alcohol fermentation. The CO2 being produced is what's causing the bread to rise and all the little air pockets in the bread to form. Now, as soon as you stick the risen bread dough into the oven, it will quickly kill the yeast and stop the fermentation process. But the bread kind of gets locked in that form and baked. All right, now lactic acid fermentation. Again, now here we're taking the pyruvic acid, we're making use of electron carriers, and this time producing lactic acid. There's no CO2 produced with lactic acid fermentation. We make use of this in the production of certain types of foods, in particular yogurt. Again, there should be an arrow here. I'm not sure why there isn't. So you take the pyruvic acid, you use those electron carriers, you produce lactic acid and NAD. And so as the process proceeds, you'll notice what's happening. In particular, lactic acid organisms make use of not glucose exactly, but a variant of that known as lactose or lactose sugar, which is what you find in milk, the kind of sugar you find in milk. And so over time, the microbe is consuming the sugar, its population will grow, and the amount of lactic acid present will increase over time. This is what gives plain yogurt the sort of sour flavor that it has, sour taste that it has, is the lactic acid in there. And of course, we often add sugar and such to reduce that sourness, that tartness. Lactic acid fermentation also happens in us when we have an oxygen debt in our muscles, when we're exercising vigorously. And it, as we probably all experience, can cause cramping of those muscles because what's happening is instead of doing cellular respiration, you're doing lactic acid fermentation instead. The lactate begins to build up in your muscle, which causes that cramping and burning sensation in your muscles. Now, what your body does, of course, that cramping and the pain is a signal to your body to, or signal from your muscles to sort of take it easy. But then shortly after that, the lactate is removed from those muscles. It's taken to the liver, where essentially the liver deals with the lactate, turning it back into glucose, essentially. And this is a process known as the Cori cycle, again, involving your liver to essentially detoxify, remove that lactic acid from the system because you don't want it building up too much. Now, of course, you have sources of energy available in your body. You have a lot of these sugars that are stored. You have them constantly being transported to the cells of your body to be used to produce ATP as a source of energy. And so this is just kind of always happening. And even when you're inactive, it's happening at a low level. And so there's always some ATP in the cells that's available to do work. So when you have short-term energy needs, and so the book talks about sprinters, for example, they need energy really quick. They're able to make use of the ATP that is in your muscles and they can take advantage of essentially fermentation or lactic acid fermentation as a short-term method to produce ATP, again, not using oxygen. And for a sprinter, that's really all you need to take advantage of is the ATP that's already there and what you can generate through lactic acid fermentation. Then, of course, the sprinter's done and they breathing heavily, getting oxygen in, dealing with that lactate, and they recover. Now, longer term energy needs. So sprinting is not an activity that you can keep up for very long because, again, you can't get oxygen to your muscles quickly enough. You do lactic acid fermentation, the lactate builds up and it starts to cause problems in your muscles. But if you exercise at a somewhat lower level, you can keep up that activity level for longer terms because there, essentially, if you're a marathoner, for example, you're not sprinting, you're running, but not at an extremely fast pace. And there you essentially make use of cellular respiration to supply the ATP that you need. And that's something that, again, at a lower level of activity, cellular respiration is fine. You can get oxygen to your cells quickly enough to be able to do that and produce the ATP that you need. Now, I want you to contemplate for a moment the difference here. So with fermentation, whether it's alcohol fermentation or lactic acid fermentation, we're doing glycolysis, and from each molecule, C6H12O6, what am I doing? Yeah. You are essentially producing a net gain of 2 ATP as we saw. But with cellular respiration, we're able to completely break down that glucose, and we are able to make approximately 36 ATP, more or less. So you can see cellular respiration is a lot more efficient at extracting energy from the foods we eat than ferment. Actually, correction, I'm going to change it. That's about 32 ATP from the complete breakdown of each glucose. Now, the glucose molecule contains in total about 686 kilocalories of energy per mole, okay? It takes approximately 7.3 kilocalories to convert ADP to ATP. So essentially it takes about 7.3 kilocalories to make each ATP molecule. I want you to try before class, and we'll talk about this in class, to figure out essentially how efficient fermentation is at converting the energy into glucose into the energy in ATP, and then how efficient cellular respiration is at converting the energy in glucose into the energy in ATP. See if you can figure out what those efficiencies are, and we'll talk about that in class. Okay, that's it.